before we start on the program and the fellows talks that are listed in your book, we have a brief presentation from a representative from Merck.
Thank you very much. So my name is Felix Ekwamwa, and a representative for Merck for West Africa. And I already got a question about Merck, as in everybody knows Merck as a pharmaceutical company. So that's more or less like the umbrella. And Merck is the oldest pharmaceutical and chemical company in the world. It's about 350, since 1668, almost 350 years old. Yeah, exactly, yes. We are present in about 66 countries with over 50,000 employees. We invest heavily in R&D, so about 1.7 billion euros in 2015. So I'll just give you a brief overview of the various categories or the various divisions within Merck. Basically, the healthcare, which is the most popular. So our one product that almost everybody here knows is 7Cs. So if you know about 7Cs, then that's a Merck brand. Then we have the life science. So basically, this is more or less of interest to you. The life science has to do with tools and laboratory supplies for research. And then the last division is performance materials, so liquid crystal. So once you have a mobile phone or a laptop, you have a Merck in your pocket. So these are the various categories or the various divisions of Merck. Again, we have Merck, we have Millipore under Merck, and then just recently we acquired Sigma Aldrich. So it's a very huge company, a global company. We just came to Ghana just about 2 years ago supporting marketing activities. So we work through distributors like Wactec and Savaco and then PKF. So just a few slides on our life science division. We have over 300,000 products with over 600,000 SKUs, over 19,000 employees, and we are present in 66 countries. As I mentioned earlier, we just came to Ghana about 2 years ago, and so far, so good. We are very good friends with Wactec and then Noguchi as well, reason why I'm here today to support this program. So just under the life science division, we have our research solutions, we have our process solutions, for instance, the pharmaceutical ingredients, the active pharmaceutical ingredients as well. And then we have our applied solutions, so everything from flow cytometry to cell analysis to Western blotting and all these things fall under our applied and research solutions. One cutting-edge technology that we have is the Muse equipment, which we are yet to introduce onto the market. So we are not just here to sell products, we also do a lot of corporate social responsibility. So for instance, if you go downstairs, you see a pull-up that's actually calling for abstracts for young scientists. So once you go down again, you can just take the email address, and then if you have any abstract that you think you need sponsorship or a scholarship for it, we are really ready to support you as well. Again, we have something called the mini labs, which we give to, we've given some to Food and Drugs Authority and Standards Authority to help check counterfeits. We also sponsor students. So as I speak to you currently, we have about 3 students from the University of Ghana, Central University, and then Kwame Nkrumah University of Science and Technology in our R&D department in Germany, and are going some training. So this gives you just a brief overview of Merck and what we do currently in Ghana. So thank you very much for your attention. Good.
All right, so now we'll start the next session of Fellows Talks, and let me introduce our 2 chairs. Dr. Seth Awasu Oji and Asmoa Kusi.
Well, that's great introduction. For the Ghanaians, the right pronunciation is Oswedi. But I'm commonly called OA. So you can call me OA, and that should be nice. Greetings from Kintampo. I think I joined you yesterday, and been following through to this afternoon. Very exciting presentations, and I'm extremely privileged to co-chair this session with Dr. Semakusi. We will make it as lively as we can, but a lot will depend on you. By my watch, 2:15, and we are supposed to finish this by 3:30. It means we have 1 hour 15 minutes. Is that right? We are going to divide this amongst 6 people, and so you can't go more than 10 minutes. If you want to have room for any suggestions, when I was a student, I like to talk briefly and get comments from people. That helped me to shape the work I do. So keep to your time. I'm very strict. As a chairman, I'm supposed to be sitting in a chair. So there's a fine for you if I have to stand up, and because I've lost my chair, isn't it? Thank you very much. So I will take the first 3 presentations, and Dr. Asmakusi will take the remaining 3. I will introduce the topics. So if going in the order, if we have, is it Suale Majid around, please? Your time starts from now.
Good afternoon to you all. My name is Majid, and I'm presenting on the topic serum biochemical parameters and cytokine profiles associated with animal African trypanosomiasis in naturally infected cattle in Ghana.
So down one, yeah, 2.
Okay. So African trypanosomiasis is a disease that's caused by several species of extracellular protozoan parasites belonging to the genus Trypanosoma. So this image shows a Gemsa-stained slide of the bloodstream forms of the parasites. And as you can see, the parasites are distributed extracellularly in between the blood cells. So the disease is transmitted mainly cyclically by the testafly, but some species may be transmitted by other biting flies, such as the horsefly. So the disease is a disease of both humans and animals. And with the human infection, about 60 million people are at risk of this disease in 36 sub-Saharan African countries. And in recent times, there has been a decrease in the number of cases of the disease. Between 2000 and 2002, over 70% reduction in number of cases was reported. And the number of infections has even decreased more remarkably in our part of the country. In Ghana, there has been no reported case in a decade. However, with the animal infection, still remains a major constraint to livestock production in sub-Saharan Africa, causing an estimated loss of about $1.5 billion in cattle production alone. And in Ghana, it's also a major problem, as prevalence has been shown to range between 5 to 50%, moving from non-endemic areas to endemic locations. And infection causes the disease animal trypanosomiasis, which results in several clinical manifestations, including what is observed here, severe weight loss. And I believe most of our faculty and then the invited guests were having barbecue yesterday, and most of you might have tasted beef kebab. So if we don't tackle a problem like this, I'm sure maybe the next time we invite you, we'll not be able to serve you very nice beef kebab. So serum biochemical aberrations and variability in cytokine profiles have also been shown to occur once there is infection. However, these studies were mainly cross-sectional studies, And also there were experimental infection with only a single species of trypanosome. And this does not reflect what actually happens in the field, where organisms are infected with trypanosomes of different species and in some cases mixed infections. So this led me to the aim of the study, which was to characterize animal African trypanosomes and determine the associated serum biochemical parameters and cytokine profiles in naturally infected cattle in Ghana over a 6-month period. So I designed a longitudinal study to take care of the cross-sectional study that were carried out. So to achieve this, we selected 2 study areas, one in Accra and the other in Adidomi. These 2 areas differ in the testfly populations. Accra has a low burden of testfly, and Adidomi has a high burden. Accra also has a low prevalence of trypanosomiasis, while Adidomi has a very high prevalence of trypanosomiasis. And so 20 cattle were selected randomly from each of these study sites, and samples were collected from these cattle, blood samples, at 6 different time points at approximately 5 weeks intervals. Then the samples were transported to the lab where we extracted DNA and then separated the serum. And with the characterization of the trypanosomes, we did a nested ITS-based PCR. This is a multi-species detection technique in that the internal transcribed spacer region is conserved within species but has interspecies variability. So just by performing the PCR, we are able to identify different species of trypanosome based on the PCR product length. But I went ahead to sequence the unique amplicons that were obtained to confirm if these species were really the species of trypanosome. And with the serum biochemical parameters, it was measured using the automated clinical chemistry analyzer, and then we did a sandwich ELISA assay to quantify cytokines. And this is a representative gel for the infection, and we found that as we were expecting, we had different band sizes for each of the trypanosomes. So for example, this band correspond to a band size for Trypanosoma vivax. We had one that is for Trypanosoma theileri, and where there are mixed infections, we obtain different band sizes for that. So this is a summary of the species that were characterized from both locations and at each of the time points. So we saw a variability in the dynamics from one time point to the other, looking at the species. And Accra had predominantly T. theileri infections, whilst in Adidomi we had T. Trypanosoma vivax being the predominant infecting trypanosome species. And these differences in the predominant infecting species was attributed to differences in the burden of testflies, as Adidomi is known to have very high testfly density. And Trypanosoma vivax is a heterogeneous parasite, means it's able to be transmitted by both mechanical means and cyclical means, meaning if we have more testflies at this location, then there's going to be a higher transmission of these parasites in those locations. So in order to determine the effect of the infection on the serum biochemical parameters, we wanted to take off some confounding factors. So we looked at other factors that could affect the levels of the serum biochemical parameters. So we look at the age and then the breed of cattle and also the geographical locations we were studying. With age and breed, we found no significant effect of these on the biochemical parameters. But with geographical location, we observed significant differences at each of the time points we studied for 5 out of the 7, and for 2 of them we found at 3 time points there were significant differences in the levels of the serum biochemical parameters. So this just goes to show that there is variability in the serum biochemical parameters between animals in the different locations. So we went ahead to look at infection, analyzing them for each of the study locations. And we found that in the Accra study area, there was no significant difference at most of the time points. Generally, the serum biochemical parameters were similar between infected and uninfected cattle. And this was attributed to the high prevalence of Trypanosoma theileri. This is a relatively non-pathogenic trypanosome parasite, and so its presence in the animals may not cause any significant pathology associated with trypanosome infection. Also, Trypanosoma theileri is able to progress to chronic infections in animals, and with chronic infections, usually in early chronicity, serum biochemical parameters fall down to near normal values, and so this might also account for this observation. The story was different at Adidomi, where we observed differences in 5 out of the 7 biochemical parameters at least one time point. And this is because this area is a high transmission area and has trypanosoma vivax infections there. So we believe that there might be there might be repeated infection with different parasites in this population, and due to the pathogenicity of the vivax infection too, it might be causing these changes we are observing here. So we went ahead to look at the cytokine profiles. So comparing the levels of the cytokines we measured between infected and uninfected cattle, we found elevated levels of interleukin-10 and TNF-alpha in the infected cattle. And this particular cytokines, their roles have been shown. They have been shown to play critical roles in trypanosome infection. TNF-alpha, for example, mediates parasitemia, and interleukin-10 is able to downregulate excessive pro-inflammatory response. And having very high levels of interleukin-10 relative to TNF-alpha shows that we have a high anti-inflammatory response in the infected cattle in this location. Comparing between the two locations, we saw that this was still the trend. In Accra, there was this high anti-inflammatory response. Then we went ahead to look at the cytokine ratio, since this would give us a good predictability of the type of immune response that is dominant. And we found that both at basal levels and during infection, we had high pro-inflammatory response, as the pro to anti-inflammatory cytokine ratios were significantly higher in adidome. So you realize that with infection and at basal level, we still had high pro-inflammatory response. And this is supported by observations we made in cattle that remained infected throughout all the time point. We saw that there was an increasing trend in the levels of the pro to anti-inflammatory cytokines, suggesting that as the infection persisted, we were still having a high pro-inflammatory response, as opposed to these two, which are from the Accra study side, where there was a decreasing trend in the pro to anti-inflammatory cytokines, suggesting a higher anti-inflammatory response here. This is wasn't what we expected, because we expect that if there is an infection and it persists for long, you should have anti-inflammatory response to counter any pro-inflammatory response you observe. But then we believe this trend we are observing here is because these animals were infected at different time points with different types of parasites, different species of parasites. And we believe that because trypanosome species also express different VSGs, it's possible that there are different antigenic types exposed to the animals at each of the time points, and this might cause the high pro-inflammatory response that is occurring, as opposed to here where we had T. tyleri infections throughout all the time points, and so there might be just one single antigenic type that is exposed to the animals. So by way of summary, T. tyleri and T. vivax were the predominant infecting trypanosome species at Accra and Adidome. And also the geographical distribution was found to influence the levels of serum biochemical parameters. Infected cattle in Accra generally had similar serum biochemical parameters as uninfected controls. And also we observed significantly elevated levels of creatinine, ALP, total protein and total bilirubin, and significantly decreased levels of cholesterol. Cattle at Adidome also had a high pro-inflammatory response, while infected cattle at Accra had high anti-inflammatory response. So from this data, we found that there was a more complicated host pathogen interaction with natural infection compared to what was reported looking at the experimental infections that were made. So I would like to acknowledge the following institutions for funding, my supervisors, and then the Trypanosome Research Group at WACBEP. Thank you.
Well done, Majid. I think the presentation really flowed. You really knew what you were talking about.
Thank you.
But the most important thing I picked is that this evening I'll look out for beef. And make an informed decision whether to eat some or not.
Well, it's nothing to really be scared about.
Oh, okay.
What we are concerned about is if we do not tackle this or control this, we'll run out of.
I just made that. Thank you. Please, any questions? Any clarifications? Yeah, okay.
Your ratio with TNF, did
Yeah, I did a TNF. Yeah.
Ratio. No, the ratio.
The ratio.
2 out of 10.
There was one.
Yeah, that one wasn't significant. TNF out, yeah, okay. There were some.
Yeah.
What was the difference between infected and uninfected?
With TNF, yeah, there was a difference.
Where's that one?
Yeah. With the ratio, no, but when I did comparison, just with the raw values between infected and uninfected, there was significant difference observed. Yeah.
Did you normalize prior to doing the ratio?
Well, when we generated, we tried normalizing, but still the data wasn't normally distributed after we log transformed the data. So I had to just use a non-parametric alternative to do the comparison. Yeah. So I used the medians instead of looking at the mean.
Any idea what actually causes the muscle waste in these animals?
Well, recently there have been reports of the parasite sequestering in certain vital organs, and this could be a result of that. But I don't know of any study that has proven this. Appetite. Oh, no, it doesn't. No, it doesn't. And the animal, no, and the animal forms of the, yeah, the animal forms of the parasites, they do not infect humans because of the trypanosome lytic factor. So we have an. Oh yes, it does. It does. It leads to weakness and then. Yeah, they don't eat. Yeah, it does. It does.
Okay. Thanks very much, Majid. So we'll move to the second presentation. To help you, I will always raise these two fingers. In my tradition, when they do that, it means you're a big man talking and people are according you respect. So watch my two fingers. It means 2 minutes. Okay, and you are not obliged to answer any question that the chair does not give permission. So all the people asking questions should address me. If I don't give you a chance, don't talk.
Good afternoon. I'm Emmanuel Amlavu. I'm working under Dr. Gordon Awandere. I'm here to present a paper titled GPI-linked SERPA TEDAS PF-RH5 PF-RIPAR complex on the merozoite surface during erythrocyte invasion. Erythrocyte invasion is a complex process that involves ligand receptor interactions and protein-protein interactions. It begins with a low affinity binding that involves soluble and GPI-anchored merozoite surface protein. This step is followed by apical reorientation that eventually leads to the formation of a tight junction. The formation of a tight junction signifies the parasite commitment to invade red blood cells, and at this point, EBL and RH families of proteins are recruited. The tight junction eventually moves preferentially around the merozoid, resulting in invagination and shedding of the dense surface merozoid coat. The merozoid is then encapsulated within the red blood cells, and then there's a brief period of echinocytosis where there's a spike in morphological changes, and then the parasite regains that, and then the cycle continues. Now, in an effort to identify novel parasite proteins that are involved in erythrocyte invasion, we had considered some selection criteria that involve resource we obtained from Plasmodb and using some bioinformatic tools and published transcriptome and proteomic data. We identified PFD1130W, a protein that has got a signal peptide, a von Weisbrand factor, and predicted to have a GPI attachment site, and a sub-1 cleavage site somewhere close to the amino terminus. Now, secondly, we also noted that PFD 1130W is located in chromosome 4, a region we termed the invasion locus because PFD 1130W is here, and just below that you have PFRH5 and PFRH4, which are well-characterized invasion type genes. Now, we need reagents to be able to properly characterize this protein. And then the first thing we did was it's a 2-exon gene actually. We made the region upstream and downstream, as well as the matured protein devoid of the signal peptide. This is just a reflection of the recombinant proteins that were made, and then the detection by anti-His molecular antibody. Now, while this study was on, we got one of the greatest shock by another group in Switzerland that immediately published this same candidate as a vaccine candidate. So while we were repositioning on how to proceed, we took up a second objective, which is to address how PFRH5, a soluble protein, how it gets trafficked to apical organelles. Alan Commons' group at WRC published that PF-RIPAR forms a complex with PFRH5, and in our own group, we were at that time characterizing PFRH5. Unfortunately, these two studies could not address the existing scientific conundrum, given that PFRH5 lacks a transmembrane domain and a GPI anchor. As such, we could not attribute any explanation as to how this PFRH5 gets tethered on the merozoite surface. Now, in order to do this, we conjectured that PFRH5 could be functionally dependent on an interaction with another protein, which may facilitate successful organellar targeting. So simply, we conducted blue native page electrophoresis and found that native PFRH5 migrated at about 200 kilodalton, which ordinarily under denaturing conditions should migrate at about